Hello, in this video we will discuss about the amyloidosis, the systemic amyloidosis. So in this way, what is this? Basically, this is the misfolding of the protein will lead to damage our tissue, damage our organs. So let's begin to understand that it is caused by misfolding of the protein. Misfolding means the correct, not correct folding, and extracellular deposition of circulating proteins. So the circulating proteins as amyloid fibrils, resulting deposition resulting in dysfunction of vital organs. So the dysfunction of the vital organs will be occur due to these amyloid fibrils. So anyhow, the dendritic cell, let's begin to understand the immune response. So here is the dendritic cell which that carry the antigen to show the antigen presenting cell through MSC class 1 and CD4, and in this way the CD4 positive T helper cell which that recognize the naive B cell, and the cytokines will produce to recruit more naive B cell in the lymphatic system. So dendritic cell will go into the lymphatic system because the T cell and B cell are present in the lymphatic system. So the naive B cell will produce the B memory cell for again infection to prevention, while the plasma cell which that is produce a lot of antibody during the infection time when the at the spot of antigen presentation. So in this way, here is the transcription factor will bind with the DNA of the plasma cell. So this is the DNA damage due to epigenetic modification or other things will lead to transcription factor to produce messenger RNA. But this messenger RNA will produce the normal antibody as well as if the damage, it will produce light chain protein, not like here antibodies. So these antibodies basically can against this light chain protein, but sometime no restriction. So the restriction will not occur. In this way, the light chain protein will grow. So important things you should need to understand on the microscopic study, the staining with Congo red dye of this light chain protein shown in the diagram. So important things you should need to understand that when vaccination to prevention by again this antigen to boost antibody due to the boosting of antibody which that is against this light chain protein to prevention that disease. But in this case, when the initiation of the light chain protein formation which can be prevented through vaccination. So anyhow, here is the amyloid deposits are widespread and typically accumulate gradually, continues to be a fatal, and is responsible for about 1 in 1500 deaths per year in UK. So this is a study showing. So let's begin to understand this is the amyloid which that is the vaccination to prevention by again this antigen to boost antibody. So to boost antibody which that will prevent this amyloid fibrils. But this long term production of the amyloid which can cause the death and which can cause the dysfunction of the tissue and dysfunction of the organ. So here is the process of the plasma cell, which that protein synthesis, how it's protein synthesis. It will produce the messenger RNA through transcription by the adding of the transcription factor, and in this way the transcribed messenger RNA, which that is the siRNA, antisense oligonucleotides, which that is basically bind here and to endoplasmic reticulum to synthesize the protein, and processing of that protein will be exocytosed, and this protein basically should be folding correctly in the Golgi bodies and endoplasmic reticulum. This is the protein synthesis, but in this case the misfolding will be occur. The misfolding protein due to the DNA damage or the subunit of the amino acid problem. So here is the siRNA antisense oligonucleotides can inhibit this production of protein. So this is the misfolding protein due to some process and some reason. So in this way, here you can see this misfolding protein will extracellular fibrillar protein will become. After this, basically the protease inhibitor, so the proteolysis will be occur. This proteolysis will produce the oligomers, and this oligomers basically is harmful. So remember, this is the small molecular drug extracellular chaperone which can be inhibit this misfolding protein, but when it will go and not inhibit, so the proteolysis will be occur. This proteolysis basically after the proteolysis to produce oligomers, but the protease inhibitors can inhibit this formation of oligomers. So the oligomers is basically will convert into the glycose aminoglycans, and these oligomers will target to the different tissue to cause the dysfunction. But here you can see this the green color showing the amyloid deposit. This amyloid deposit is basically green color, so that is why it's known as amyloid. And this amyloid deposits which that is basically CDHPC and MAB, anti-amyloid MAB, isodoxorubicin, and the doxycycline, which that will be inhibit this green color showing that amyloid deposit inhibition. But in this case here you can see the amyloid deposit can be can create several function. So here is the glycose aminoglycans which that will convert into the amyloid deposit, but the GAGs mimetics can be inhibit this process oligomers to the amyloid deposit. So the deposit can inhibit. While here the CPHTC can also lead to produce the SAD and which that will create more amyloid deposit. So the amyloid deposit, angiogenesis, oxidative stress, and tissue and organ cell injury will cause these things. So the causes of the risk factor alcohol, medication, and smoking, which that will lead to change the plasma cell DNA, the DNA, epigenetic mutation or radiation or aging and gene mutation. In this way, the bone marrow containing the B cell, which that will proliferate into the B memory cell and plasma cell. So this is the immunoglobulin light chain will produce also as well as the antibody. So the immunoglobulin light chain and antibody complete immunoglobulin will produce, but the immunoglobulin light chain is harmful because the immunoglobulin is very important for combating and against the antigen, but this light chain is not any function. So the immunoglobulin light chain here is produced, which that convert into, again I am discussing the monomers and ultimately oligomers and amyloid fibrils again. So here the liver produce the transthyretin. This TTR transthyretin stabilization, transthyretin basically will convert the monomers from the light chain. So these monomers basically convert into the oligomers by several function, and this amyloid fibril will be formed. And this amyloid fibril in the presence of the human growth hormone, the human growth factors, which that will lead to angiogenesis, the increasing of the blood vessel in the unneeded area of the tissue and organs. So in the other hand, here is the basically is the oxidative stress, inflammation, and uric acid increase due to these monomers. So anyhow, this myeloid fibril will lead to fibrosis in the several tissue and matrix deposition, also the fibrosis in liver, and on the other hand, the dysfunction of organs ultimately lead to death. So this is the amyloidosis, which that is the risk factor: alcohol, medication, smoking, or other epigenetic mutation. As well as on the other hand, there is several function, the aging, which that will lead to cause the amyloidosis. So I hope you make sense about these things. This video about the overview of the amyloidosis, a systemic amyloidosis. So thanks for watching. Bye. Please make sure to subscribe, like, and share.